Welcome to the GobyWeb video training series. In this tutorial, we will demonstrate how to carry out a differential expression analysis from RNA-seq data. For this tutorial, we will be using a publicly available dataset from GEO, the Gene Expression Omnibus, accession number GSE33141. In this study, wild-type mice retina were compared with neural retina-specific leucine zipper double knockout mice, NRL double knockouts. We have 3 biological replicates for each condition. These samples were previously aligned using GobyWeb, and we will now show how to carry out the differential expression analysis. Navigate to the Actions menu and select Compare Groups of Samples. The first step is to define an overview of the comparison. We give the comparison a description. In the analysis configuration, we describe the analysis type. Here we want to carry out differential expression analysis with Goby. We want to compare 2 groups of samples, and the organism here is mouse. The reference genome we aligned against is MM9. We select that here. There are other configurable analysis options for differential expression analysis with Goby. You can opt to use weight adjustment. This applies to applying bias correction, for example, for GC content biases for the specific samples. Annotation type, yes, we're interested in comparing genes, so we select that here, and again here, genes specifically. In the advanced configuration, you can opt to include reference names. That is, if you want to perform the differential expression analysis for a subset of chromosomes, you can select that. So here by giving a comma-separated list. And then the next options are for sharing. You can opt to share the differential expression analysis here. The next step is to define the first group. We click Define Group 1. This takes the wizard to the next step. The description and analysis configuration are already set. We give a group name for that group. The next step is to select the alignments for the samples in that group. The next step is to define the second group. We give a meaningful group name for the second group. We select the alignments for that group. And select Verify Differential Expression Analysis. This takes us to the final step, the verification step. Here you check that your configuration is set up correctly and that you have the right alignments defining each group. We then select Save Differential Expression Analysis. The differential expression analysis job is created. And you see, once again, the processing status and status history boxes, which allow you to keep track of the job. To check the status of the differential analysis job, you navigate to Browse and select Differential Expression Analyses. Here you'll see a list of differential expression analyses. The job status for a wild type versus in-around knockout is completed. We select the job. This takes us to the Show Differential Expression Analysis page. The top half of the page shows you the configuration for the job, and the job status shows completed. The completed analysis menu here, you can choose to browse the differential expression analysis results or download the full tab-separated file for the job. You also get an analysis plot. Here it's a scatter plot showing log2 RPKMs in the wild type group versus the knockout group. You see here we have some differentially expressed genes here and here. The status history gives you a picture of how long it took for the job to complete and when each task was carried out. So here we navigate to Browse Differential Expression Analysis Results. This gives you a dynamic table that you can query for the different genes. The Browse Differential Expression Analysis Results page allows you to look at your results, filter on particular columns, sort, and reorder the columns, and export different views from GobyWeb. If you click at the bottom here, Columns menu shows you a list of all the columns that we have for this table. We have the element ID, in this case is the gene ID, RPKMs for each of the 6 samples, log2 RPKMs, the counts for each sample, fold change, average RPKM across groups, t-test statistic for comparison between wild type and knockout, as well as Fisher's exact test, p-values, and false discovery rate implementation of Benjamini-Hodgberg correction for the p-values. So you can select to remove all the columns from the table. You can add all of them. You can add a subset. For example, let's say I wanted to just look at RPKM for all 6 samples. I would select OK. This would then give me a table with just the gene IDs and those 6 columns. And I could export this view of the table to a tab-separated file, and I could analyze that further using other tools. I could use Excel or R or any other TSV reading software. Another useful, let me add all the columns back. Another useful tool here is to filter this table. If we want to know which genes are significant, we navigate to the column with the Fisher's exact test p-value corrected. So here we have the Benjamini-Hodgberg q-value. We want to know how many genes had a q-value less than 0.05. So we filter on that box, and here on the bottom right-hand corner, you see now the gene count is reduced to 11,400. We could further filter the table based on fold changes. The column here that shows the fold change magnitude, that is in both directions, fold change in both directions. So let's say we then say filter and give me the number of genes that are greater than fold change of 2, over or underexpressed. Again, we see the gene count is reduced to 563. We can opt to export the subset of the differential expression analysis table of only the significant genes. You would click export, and here you would see only that table you will only have 563 rows in that table. This is useful sometimes because you can have millions of very large tables that become unmanageable. So it's useful to be able to perform this filtering. Once you're finished with this, you can click Clear Filters, and this will give you the original table. This concludes the tutorial on differential expression analysis. For more information, please visit our website at GobyWeb.campagnalab.org.